Hello student, good afternoon. Today we will start the next topic, that is endoplasmic reticulum. Structure of endoplasmic reticulum. The cytoplasmic matrix is the traversed by a complex network of interconnecting membrane bound vacuoles or cavities. The vacuoles or cavities often remain concentrated in the endoplasmic portion of the cytoplasm, therefore known as endoplasmic reticulum. The endoplasmic reticulum was first of all reported by Porter in 1945. Porter scientists have described the in 1945. Fawcett and Ito have studied the 1958 theory in 1958, and Rose and Pomeroy in 1960 have made various important contributions of the endoplasmic reticulum. All these scientists have studied the different assumptions on the endoplasmic reticulum. The endoplasmic reticulum varies from cell to cell. Endoplasmic reticulum varies from cell to cell. The egg and embryonic cells lack in endoplasmic reticulum. Egg and embryonic cells lack means egg and embryonic cells have absent endoplasmic reticulum. The spermatocyte have poorly developed endoplasmic reticulum. Spermatocyte have poorly developed endoplasmic reticulum. The adipose tissues, brown fat cells, and adrenocortical cells of opossums testes contains only smooth or a granular type of endoplasmic reticulum. The cells of those organs which are actively engaged in the synthesis of proteins as the pancreas and other endocrine glands are found to contain highly developed endoplasmic reticulum. Endocrine glands and found the contains highly developed endoplasmic reticulum. Most endoplasmic reticulum is of granular type. Two types of endoplasmic reticulum, granular and agranular. Granular means rough endoplasmic reticulum. Agranular means smooth endoplasmic reticulum. The liver cells possess both the types of endoplasmic reticulum. In the liver cells, two types of both the type of endoplasmic reticulum, that is granular as well as agranular. Structures. Morphologically, the endoplasmic reticulum is composed of following three kinds of the structure, namely these are as follows: cisternae, vesicles, and tubules. All this combination to make the endoplasmic reticulum: cisternae, vesicles, and tubules. Number one, cisternae. These endoplasmic reticulum cisternae are long, flattened, sac-like, unbranched tubules. These cisternae having the diameter of 40 to 50 MV. They remain arranged parallelly in bundles or stalks. The cisternae usually occurs in those cells which have synthetic 40 to 50 mu. They remain arranged parallelly in bundles or stalk. The cisternae usually occurs in those cells which have synthetic roles as the cells of the pancreas, notochord, and brain. Cisternae components of endoplasmic reticulum. In this cisternae pre-ribosomes and granular ribosomes. Vesicles. The vesicles are oval membrane bound vascular structures. They having the diameter of 25 to 500 micron. They often remain isolated in the cytoplasm. Present in most cells but especially abundant in the pancreatic cells. Vesicles are present. Vesicle components of endoplasmic reticulum. Tubules, cisternae, vesicles, and third is tubules. The tubules are branched structures forming the reticular system. Along with the cisternae and vesicles, they usually have the diameter from 50 to 190 micron and presents almost in all the cells. The cavities of cisternae, vesicles, and tubules of the endoplasmic reticulum are bounded by a thin membrane of 50 to 60 angstrom unit thickness. The membrane of endoplasmic reticulums is trilaminar like the unit membrane of the plasma membrane, nucleus, Golgi complex, etc. The membrane thus is composed of an outer and inner dense layers of the protein mold. The cavity of the endoplasmic reticulum is well developed and acts as the passage for the secretory products. Palade 1956 observed secretory granules in the cavity of endoplasmic reticulum. This is the structure of endoplasmic reticulum. These types of endoplasmic reticulum is to start the next period. Okay, thank you.